Muito bem. Bom dia a todos, bom dia a todas. Vamos dar início, então, ao nosso Simpósio Internacional de Metabolismo e Câncer. Eu vou falar rapidamente antes de iniciarmos o evento e, a partir de então, eu também vou alterar para o inglês, já que é um simpósio internacional e temos convidados palestrantes estrangeiros. So I would like to welcome all. We are discussing one of the most important hallmarks of cancer that has been recognized in the recent years. Our international symposium will be very focused on that. For that, I would like to thank in advance all the speakers that accepted to join us today and tomorrow. I would like to thank also the organizers of this meeting, Dr. Luiz Gustavo de Bois, who conceived this symposium, Dr. Gabriela Nestal and Dr. Vanessa Sandin, who are also involved in. And I would like to promote our annual symposium in oncobiology that is going to happen next November. For those who want to assist some interesting speakers, we still have time to join. So, coming back to the program, we can have today 3 speakers, and we can have 3 speakers. The first one today is Dr. Alicia Kowaltowski. So I'd like to thank in advance Dr. Alicia for joining us. So Dr. Alicia is graduated in medicine at the State University of Campinas, Unicamp, in 1997. Under the supervision of Dr. Aníbal Versesi, Alicia defended her PhD in medical science at the same university in 1999 and further did a postdoc at the Oregon Graduate Institute in 1999 and 2000. She's currently a full professor at the Department of Biochemistry at the University of São Paulo, and her research group is interested in the field of bioenergetics, transport, and mitochondrial redox state. So I would like to thank Dr. Alicia for accepting the invitation and to give some minutes to us to talk about her work, what is more focused on metabolism, I may say. And Alicia, thank you so much.
So good morning. Thank you for the invitation. I have to thank Luiz Gustavo, Robson, Gabriela, and Vanessa for organizing this. It's really nice to be speaking here. Let me try to share my screen and let me know if you have any problems. Do you see my screen? Well, if somebody has some kind of problem and can't see me, please interrupt me. I think I'm sharing. So as I told Luiz Gustavo and Robson when I was first invited to give this talk, I work with mitochondrial energy metabolism in the lab. And we actually spread our research areas very thin in the lab and study many different aspects of mitochondrial energy metabolism. But the one aspect of energy metabolism I do not study is cancer. And that's not because I don't think cancer is interesting or important or anything like that. It's just that I think that would be really spreading too thin for my lab. As far as I can tell, cancer is extremely complex in terms of metabolic plasticity. It's also very specific to different types of cancer, and it's just too much. I can't really follow the literature in cancer research. So I focus on mitochondrial energy metabolism in basically everything but cancer. And when I warned the organizers about this, they said, it's fine, you can talk about whatever you want. So I do have to warn everybody who's here for a symposium in metabolism and cancer that you're going to get a lot of metabolism in this talk, but not any cancer whatsoever, because I don't do any cancer, and I'm sorry about that. Having said that, I tried to think of something that we are doing in the lab right now that could interest cancer researchers. And what I decided to talk about is something that I don't usually do, is the specific work of one student in my lab, which is the specific work of, and for some reason the slide doesn't go, okay, now it did, which is the specific work of Pamela Kakimoto, who is a PhD student who defended earlier this year, and finally, after 6 months of limbo, managed to start a postdoc in Germany right now. And the reason I want to present her work is that because during her PhD work, and also in collaboration with Antonio Zorzano in Spain, she found that you can regulate glycolysis by fatty acids. And I think that the regulation of glycolysis and this new mode of regulation of glycolysis that she uncovered during her work should be of interest to cancer researchers, because the modulation of glycolysis is classically related to cancer research. So having said that, I'm giving a seminar that I have never given before based on the work of one single student in my lab. So please forgive me if the seminar is a little bit rough around the edges, but I hope it's interesting to this group. So the studies that Pamela was doing during her PhD were actually to try to understand what happens when you convert a normal liver to then a fatty liver, which is a liver that has an accumulation of fat in it, and that happens to all of us when we eat a meal, even just a single meal that's very rich in fat, and how this conversion can become malignant, because fatty livers are quite benign actually, become malignant in an inflammatory process in a non-alcoholic steatosis kind of model. And this conversion from a normal fatty liver to a NASH situation, a non-alcoholic steatohepatitis, is really important, because once you have this inflammation associated with NASH, then you can have a progression of liver disease, and you can have a progression to cirrhosis with liver failure, and also a progression, because of all this inflammatory damage, to cancer. So there, cancer appeared in this talk. So this conversion from normal livers to fatty livers to NASH, this inflammatory condition, was really what interested Pamela. And she wanted to understand what were the bioenergetic changes that were involved, how mitochondria were involved in this, and also how redox signaling, and particularly how reactive nitrogen species were involved in this. And basically, her question was which came first, if it was bioenergetics or if it was redox signaling, and this conversion to fatty livers into an inflammatory disease. Now, this all fits in very well with the general idea of the lab. So basically, what she was trying to understand is how mitochondrial changes were related to changes in reactive nitrogen species in the conversion of a fatty liver to NASH. So as I was saying, this all fits in very well with the general idea of the lab, which is to study molecular mechanisms behind obesity. So obesity, we all know, is a big problem worldwide. Obesity is increasing all over the world, and obesity is limiting lifespans and health spans of people, mainly in the countries that are quite developed, in which obesity is rampant. And obesity actually is decreasing life expectation in these countries. Before COVID, we were expecting already that the teenagers today in the US would have a lifespan of 5 years less than the seniors in the US because of the high prevalence of obesity. So obesity is an enormous public health problem. It increases enormously the chances of many age-related diseases, including many different kinds of cancer. But we know that for a long time, and we also know that public health education in terms of obesity is not working in curbing the obesity epidemic. So people know that obesity is undesirable from a health point of view, but they still are obese. And this is not because people are bad and eat bad food and stuff like that. It's basically because we were evolutionarily programmed to be obese. We were evolutionarily programmed to take any excess calories that we eat and deposit it as fat, And we are also evolutionarily programmed to like very high calorie foods. And the reason for this is that evolution happened over millions of years of scarcity and in which food was hard to find, and also millions of years in which the lifespan of the average human was quite low. Only in the last 100 years, when our average lifespans became over 40 and it became much easier to find food, did we start having a problem with obesity. So I don't think we're going to go any further in terms of public health and telling people how to eat healthily, because our genes are there and they tell us basically to like food and to like high calorie food. I think we have to understand mechanisms behind obesity, molecular mechanisms behind diseases that are related to obesity, in order to be able to curb the problems of obesity worldwide. In terms of obesity and in terms of high fat diets, which is what Pomela's work was related to, we normally studied these diets in animal models and specifically in rodent models. And here I think it's interesting to talk about some characteristics of rodent models that are really important when you're studying laboratory rats or mice. So one characteristic of rodent lab rodents is that we often forget about this, is that laboratory rats and mice are reared with ad libitum diets. This means that they can eat as much as they want, even though their diets are quite healthy, quite low in fat, quite high in complex carbohydrates. They can eat as much as they want, and they also don't exercise. So by the time they are adults or middle-aged, most laboratory rodents actually exhibit lots of characteristics of metabolic diseases. They're actually quite comparable to a mildly obese couch potato human. So you'll see that they have high glucose, high insulin, high circulating lipids, and also inflammatory markers that are typical of the metabolic disease. So our normal control laboratory rat or mouse already is obese and already is a model for a mild metabolic disease. So when you're studying high-fat diets, as we were in Pomela's project, you're actually studying the effect of high fat on already obese animal model. So that's something to keep in mind. And actually, if you want to compare your animals to a non-obese model, what you really should do is restrict diets or exercise these animals, because then they start looking more like a healthy human model. Also about animal models of high-fat diets, which is what Pomela was studying, an important thing to know is that the protocols and the way that they're implemented vary enormously in the literature. And Pomela was such an exceptional student in the lab that she actually went out of her way to understand how these protocols vary and understand how different protocols lead to different implementations of fatty liver disease. So she reviewed this in a structured manner, and she saw that very different diets are adopted in the literature and that they have different effects in terms of the evolution of liver disease. One point that I think is very important that she uncovered is that the control diet of the animal very often is not paired. So very often in the literature, you will find control animals being fed on normal chow, which is grain-based and quite variable in composition, while the animal that's treated with a high-fat diet has a specific composition of the diet. And she and we as a group don't believe this is a very good comparison, because then the diets are not varying only in terms of fat. What we're doing in this study is varying only the fat and carbohydrate content of the diet. So we're using known component diets, both for the control diet and for the high-fat diet. Another thing that she found is that the fat source in the diet varies a lot. It's pretty well known that unsaturated fatty acids, such as those present mainly in plant-based lipids, do not lead to as much induction of liver disease as saturated fatty acids. And actually, most diets use saturated fatty acids, such as from butter or lard. And the diet that we're adopting here is using saturated fatty acids from pork lard. So we're adopting the major fat source in the diets we're going to use. The fat composition, of course, of these diets varies a lot depending on the source of these lipids. Finally, and this is a really important point for this talk, The animal models and high-fat diets vary a lot. So if you look at rat diets, basically we use two kinds of strains of rats. We use either Worcester or Sprague-Dawley rats. But if you look at mouse strains, even though most people actually use C57 blacks, there are some other mouse strains used, there are lots of subtypes of C57 blacks. And that's really important in animal model studies, and particularly in metabolism and bioenergetics, because the major mouse group used in high-fat diet studies are the C57 black 6J mice. And what are these black 6J mice, and what are the black 6N mice? What are these mice in terms of differences in these strains? This is something that you really should pay attention to if you're studying animal models and pertaining to metabolism specifically, and specifically mitochondrial metabolism. Because the black 6J mouse has a specific characteristic about its mitochondria, which is the lack of a protein called the NNT, or the nicotinamide nucleotide transhydrogenase. So many decades ago, what happened was that within the Jackson laboratories, there was a spontaneous mutation. And because laboratory mice are so inbred, this spontaneous mutation just spread across the whole colony. And today, the Jackson laboratory mice that descend from those mice all do not have any activity of the NNT. So what is the NNT? The NNT is an inner mitochondrial membrane protein that spans the inner mitochondrial membrane and uses the mitochondrial inner membrane potential as an energy source to transfer electrons from NADH, which is present in mitochondria from, for example, the Krebs cycle, to NADP, generating NADPH. And this is very important because mitochondria don't have the pentose pathway to give them NADPH. And actually their main source of reduced NADPH is the NNT. And NADPH is very important in mitochondria because it reduces thiol proteins or thiols such as glutathione. And this is used to remove peroxides from mitochondria. So mitochondria that don't have the NNT through this spontaneous mutation actually have low NADPH, low glutathione levels, and have a defect in the ability to remove hydrogen peroxide and other organic peroxides. Because of that, animals that have this mutation are actually constantly in a state of mitochondrial oxidative imbalance. And because of that, also in the last few years, many different researchers have found that these animals are more prone to metabolic diseases. So if you're studying mice that descend from these Jackson laboratory mice, it's a good idea to check them in terms of them having or not the activity or a truncated version of the NNT, because that's going to determine if these mice are J mice, which are the ones that don't have the activity of the transhydrogenase, or if they're N mice that do have the activity of the transhydrogenase, because these different mice behave differently in terms of susceptibility to metabolic diseases. And just so you have an idea of how much of an effect an NT deletion has, this is data from Roger Castillo's and Anivo Versasi's group. Basically, what they did was look at NADPH redox state in isolated mitochondria, and NADPH is fluorescent when it's reduced. Then they oxidized NADPH with tertiary-butyl hydroperoxide, and they can see that in the J mice, which is the mice that most of us study, the recovery of the reduced form is much delayed relative to mice that have the intact NNT, so they can recover this NADPH very quickly. And in addition to that, the J mice that most of us use have more oxidized glutathione. So they're in a constant state of mitochondrial oxidative stress and oxidative imbalance. So knowing that, Pomela was studying mice that did not have the deletion of the NNT, so mice that are intact in terms of their mitochondrial redox state. And she decided to study what happened with these mice in terms of their mitochondrial profile at different times when you gave them a high-fat diet. She gave them a very controlled high-fat diet in which only carbohydrates and fat were switched in terms of content. And she compared the effects of wild-type mice and also mice lacking nitric oxide synthase, because nitric oxide from inducible nitric oxide synthase had been shown prior to the study to be involved in the generation of inflammatory forms of fatty liver disease. So she wanted to understand what came first, if it was nitric oxide signaling or if it was mitochondrial bioenergetics that was changed first in the metabolic disease induced by fats and also increased by the presence of nitric oxide synthase. So she looked at the changes in metabolic parameters in these animals over time at 2, 4, and 8 weeks. And the first thing that she saw was that these animals gain weight on high-fat diets. I guess that's not exactly a surprise. But also reproducing data from prior work, the animals that were knockouts for nitric oxide synthase actually gained a little bit more weight than the animals that weren't knockout for nitric oxide synthase. Also, not surprisingly, animals that were brought up on the high-fat diets had more fat in their bodies when we looked at their body composition. And here, really, there wasn't much of a difference in terms of the knockouts for nitric oxide synthase. She also put these animals in indirect calorimetry systems and looked at their ratios of CO2 to oxygen, which can tell us if they're using more lipids or more carbohydrates, because lipids are more reduced, so you're going to have a difference in the ratios of CO2 generated to oxygen consumed. And again, non-surprisingly, what she saw was that animals on high-fat diets use more lipids as a source of energy, while animals that are brought up predominantly with carbohydrates use more carbohydrates. This was not dependent on the presence of nitric oxide synthase. They both used more lipids, both the wild type and the knockout animals. One thing that's known about high-fat diets is that it generates insulin resistance. So she did look at insulin resistance and the effect of an insulin injection on glycemic levels at 1.5, 3, and 7 weeks, soon before she looked at the other parameters. And what she saw was that there is insulin resistance in high-fat diet animals. They have higher glycemic levels. Interestingly, this appears early, kind of disappears around 3 weeks, and then returns at 7 weeks. Maybe there's some compensatory mechanisms that after a long time on a high-fat diet, you simply can't compensate anymore. But very interestingly and surprisingly to us, the knockout of INOS had absolutely no effect on insulin responses. And this was surprising to us because prior data had shown that knockout animals for INOS had a different result in terms of insulin resistance, that actually nitric oxide synthase was important to generate insulin resistance in high-fat diets. This is another graph, another way to look at these changes in insulin sensitivity, also showing to us that we had absolutely no effect of the knockout of INOS on insulin resistance. We only had a dietary effect of high-fat diets on insulin resistance. And as I said, this was very surprising because prior to this work, a very important paper in the literature from André Maret's group had shown that nitric oxide synthase, the same nitric oxide synthase that we're using here, that we use knockout animals for, was very important in generating insulin resistance. In fact, the high plasma insulin levels seen in high-fat animals were completely absent in the knockout animals. And also those high blood glucose levels that we see in the wild-type high-fat animals, they could not see in the nitric oxide synthase knockouts. So what's the difference between his group's work and our group's work? It's also mice, it's also high-fat diets, it's also saturated fat. The main difference is that their animals were the J animals. Their animals do not have an active NNT, and therefore they do not have adequate levels, normal levels of NADPH in their mitochondria, and don't have normal reduced glutathione levels in mitochondria. Therefore, their animals are probably already in a constant state of mitochondrial oxidative imbalance. Now, this is not their fault. In 2001, very few people were aware of the fact that the strain had this mutation and had this defect in mitochondrial bioenergetics and mitochondrial redox state. I think it's really interesting because it shows that you do have an effect of nitric oxide as long as you already have oxidative stress in mitochondria. It's almost as if you needed 3 hits in terms of getting mitochondria full of fatty acids, also full of oxidative stress, and then having nitric oxide synthase, producing nitric oxide on top of that. This 3-hit system really is important in the development of liver disease. If you just have high fat, then just having nitric oxide on top of that doesn't seem to make any difference if mitochondrial antioxidant systems are intact. So this points out to a really important role of mitochondrial redox state. And in fact, mitochondrial redox state is the one thing that really changes in high-fat diets. So even prior to Pamela's work in our group, Ariel Cardoso studied the effects of high-fat diets on redox state in liver. And we can see in whole liver homogenates that markers of oxidative stress, such as carbonylated proteins, for example, increase a lot with high-fat diets. What we also see is that mitochondrial generation of reactive oxygen species, such as hydrogen peroxide, increase a lot in high-fat diets, and particularly increase when they're induced by substrates that are lipid substrates, such as palmitoyl-CoA. So you have just about double the production of hydrogen peroxide in the presence of fatty acids in animals, in the mitochondria isolated from animals that were fed high-fat diets. Interestingly, this is quite specific to very long-chain fatty acids, to long-chain fatty acids such as palmitoyl-CoA, while we cannot see a difference in the production of hydrogen peroxide when we look at shorter-chain fatty acids. So therefore, it seems to be specific to some kind of component of long-chain fatty acids. And in fact, Pamela, in the beginning of her PhD and the end also for her undergrad studies in my lab, identified the very long-chain acyl-CoA dehydrogenase, or VLCAD, which is a component of beta oxidation, the first component actually of the beta oxidation cycle, as a source of mitochondrial reactive oxygen species, as a source of mitochondrial hydrogen peroxide. What happens is that any enzyme that transfers electrons, and particularly flavoenzymes such as VLCAD, which normally reduces FAD to reduced FAD, any flavoenzyme can generate oxidants if oxygen reaches the active site of this enzyme. And what happens with the very long chain acyl-CoA dehydrogenase is that the active site is quite open because it has to receive these very large long chain fatty acids. So this active site actually permits an oxidase activity in which oxygen can be reduced to hydrogen peroxide. And in fact, when Pamela produced recombinant VLCAD, you can see the production of hydrogen peroxide by this recombinant isolated enzyme as long as you give it a substrate, so there's a source of electrons. So this enzyme is actually capable of generating hydrogen peroxide directly. Now, in animals that are fed high-fat diets, there's an induction in the production of these enzymes. So they express higher levels of most enzymes of the beta oxidation. And specifically because of that, these mitochondria, when you give them lipid substrates, generate more hydrogen peroxide. And this leads to oxidative imbalance in high-fat diets. So we know that mitochondria from animals fed high-fat diets generate more hydrogen peroxide, generate more reactive oxygen species, and have redox imbalance. What happens in terms of their mitochondrial function, in terms of oxidative phosphorylation? So to measure that, we actually use what I consider the gold standard to measure mitochondrial function, which is measuring oxygen consumption. This is the only way that you can measure mitochondrial function without having to worry about a whole bunch of artifacts that can exist in these kind of measurements. We do this with high-resolution respirometry. This is basically just a very sensitive oxygen electrode. And we can measure mitochondrial function first in the presence of ADP and phosphate, which we call state 3, which allows mitochondria to have oxidative phosphorylation, and therefore there's more oxygen consumption. And then inhibiting oxidative phosphorylation by inhibiting the ATP synthase with oligomycin. And this generates what we call state 4 respiration, which is dependent only on the integrity of the mitochondrial inner membrane. So here we can measure the integrity of the inner membrane with respiration in the absence of oxidative phosphorylation, and also the ability to promote oxidative phosphorylation in state 3. So we did these measurements in the animals that Pamela had put on high-fat diets, both the wild type and the knockout animals for INOS. And what we basically saw is that there is absolutely no effect of high-fat diets on state 3 or state 4 respiration over time. We saw a small effect of the knockout of INOS, but we saw no effect of high-fat diets on these animals. And this actually is compatible with data we had previously, in which we saw absolutely no effect of high-fat diets on mitochondrial bioenergetics with 9 months of high-fat diets. So it really seems to us that mitochondrial oxidative phosphorylation is extremely resilient in the development of fatty liver disease. And mitochondrial oxidative phosphorylation itself is not, changes in mitochondrial oxidative phosphorylation are not involved in the development of fatty livers, but maybe mitochondrial redox state is. So from the animal studies I've shown you, the conclusions we come to is that high-fat diets in mouse livers promote insulin resistance, but this is not dependent on nitric oxide synthase. It's only dependent on nitric oxide synthase if you have NNT-deficient animals, which are animals which already have a condition of oxidative stress in their mitochondria. So perhaps in these animals, because they already have high levels of reactive oxygen species in mitochondria, they might generate more peroxynitrite from the nitric oxide produced by these enzymes. And therefore, in these animals, nitric oxide is important. This also points to the fact that NNT-deficient animals, which are often used in the literature, are different metabolically. So we have to think about the presence of this enzyme when we're doing these studies. High-fat diets do not promote changes in mitochondrial oxidative phosphorylation, both short and long term, but they do produce changes in the production of oxidants by mitochondria, basically because high-fat diets induce the expression of enzymes of beta oxidation. And amongst these enzymes is the very long chain acyl-CoA dehydrogenase, which can act as a mixed oxidase and produce hydrogen peroxide itself. So this also points out to the fact that not only the mitochondrial respiratory chain can generate free radicals, but you can also generate oxidative species from other components in mitochondria. Other flavoenzymes in mitochondria are important sources of reactive oxygen species. Now, after doing these animal studies, Pamela was really interested in mechanisms of generating these fatty livers and mechanisms of lipotoxicity. And studying mechanisms in animal models is quite difficult. So she wanted to look at a cell model of lipotoxicity to see what happened in terms of metabolism in these cells, and also what happened in terms of mitochondrial size, shape, and function. So we chose a cell line, a liver cell line, which are these PLC cells, and we treated these cells with high levels of palmitate as a model for lipotoxicity, similar to what would happen in an in vivo liver in a high-fat diet. And she looked at actually, what happened in terms of mitochondrial size and shape in microscopy, and also what happens in terms of metabolism of these whole cells. And here using primarily a plated cell system to measure oxygen consumption, which is the seahorse system, and I'll show it to you in a minute. Now, one thing about lipotoxicity models in vitro is that they are not easy to do. So fatty acids dissolve poorly. You have to conjugate them to proteins such as BSA, but there are about 1 million different types of BSA you can buy from an average Sigma catalog. So if you want to know how to do all these things, Pamela and we got together actually with Oren Sherahai's group, which also had problems with this. And we wrote a review on how to do cell models of lipotoxicity, because these are not easy to do. So all the tricks, which BSA to use, which not to use, how to dissolve these fatty acids, how to calculate how much you have free fatty acid, are in this paper, which I recommend if you're good at studying lipotoxicity. So Pamela got her liver cells, and she treated them either with control BSA or with palmitate, the fatty acid conjugated to BSA, and went to the microscope to look at mitochondria. So these mitochondria were marked here. And you can see that very early on, 6 hours on, but also conserved 24 hours on, high fatty acids promote a fragmentation of the mitochondrial network. So the mitochondrial network in these cells is typically quite fused. So you have very elongated spaghetti-shaped mitochondria. And when you treat these cells with palmitate, you have much more rounded and much tinier mitochondria. This is not new. This had been seen by other authors. But basically what Pomela saw is that this happens very early on. Within 6 hours of treatment with palmitate, there's already a lot of mitochondrial fragmentation. This makes sense since over the last few years, we've seen that mitochondria are actually not individual organelles, that they're actually part of a mass of mitochondria that are constantly changing size and shape. So mitochondria, after mitochondrial components are generated through biogenesis, mitochondria are constantly undergoing fusion, in which two organelles fuse together and generate one mitochondrion, or also undergoing fission, in which one large mitochondrion is going to be separated into two mitochondria. This happens at fission sites that are marked by the ER. And these smaller mitochondria can now also change material with other mitochondria undergoing fusion cycles again. This is important to exchange mitochondrial components and to maintain a healthy mitochondrial population in the cell, because removal of mitochondrial components through mitophagy and regeneration of mitochondrial components through biogenesis depends on this process of mitochondrial dynamics. So these changes in mitochondrial shape that she saw were not surprising in this sense. Now, what do these changes in mitochondrial shape induced by fatty acids do in terms of mitochondrial function? So the first thing she looked at was mitochondrial oxidative phosphorylation in permeabilized cells. So the plasma membrane of the cells was permeabilized, so we could access the mitochondria within the cells with different substrates. And when we did this and looked at respiration in state 3 in the presence of oxidative phosphorylation, or state 4 without oxidative phosphorylation, or the ratio between state 3 and state 4, we saw absolutely no effect of the treatment of palmitate. So mitochondria in these cells have a very different size and shape. They're all fragmented, but in terms of oxidative phosphorylation, at least in the permeabilized cells, these mitochondria are working perfectly fine. So this means that not every change in size and shape in mitochondria affects oxidative phosphorylation, but it's also in keeping with the findings we see in animals in which we don't see changes in oxidative phosphorylation, even with high-fat diets, even for very prolonged periods. Now, we also wanted to look at metabolism within intact cells and not only in the mitochondria in an isolated system such as permeabilized cells. And to do that, we used a seahorse extracellular flux bioanalyzer, which basically has an electrode that measures pH and an electrode that measures oxygen, a sensor actually, it's not an electrode, that measures oxygen in a multi-well plate system. And these are mounted on pistons that actually go up and down. So you can measure this oxygen consumption and proton production and acidification from the cells with very small volumes. And what this allows for is to have a lot of sensitivity for plated cells. So the advantage of the system is for plated cells that you can have high sensitivity in terms of oxygen consumption and also parallel measurements of acidification, which allow you to do some calculations. What Pomela saw was that when she added palmitate to cells, she saw actually no difference between the palmitate-treated cells or the non-palmitate-treated cells, which are the black cells, in terms of oxygen consumption. There was a change of oxygen consumption over time of all these cells because they were put in a new system, but there was no difference between the palmitate-treated or untreated cells. And this, again, is compatible with the fact that we don't find any change in oxidative phosphorylation when we look at the permeabilized cells, when we look at the mitochondria in the permeabilized cells. Now, very surprisingly, and actually happening really early after the addition of palmitate, less than an hour after the addition of palmitate, what she very consistently saw was that the palmitate-treated cells, the lipotoxicity model cells, generated more acid. So there was more extracellular acidification with no change in oxygen consumption. Now, why were these cells generating more acid? One of the reasons can be these cells are incubated with glucose as a substrate. And if you have more production of ATP through the glycolytic pathway generating more lactate, this is associated with more production of acid or more proton release. And actually, when we measured secreted lactate from these cells, very surprisingly, we saw that there was an increase in lactate production induced by treating the cells with palmitate. So if you treat a liver cell with a fatty acid at toxic concentrations, this fatty acid induces more lactate production. We were also able to calculate the amount of ATP produced by oxidative phosphorylation and by the glycolytic pathway. You can do this with the seahorse system because you have this consumption of oxygen in parallel with the acidification. And you can also add inhibitors of different pathways and then see how these rates change. You can calculate these ATP production rates. These are published protocols that we adapted for ourselves. And very surprisingly, again, ATP production by oxidative phosphorylation was exactly the same. I'm not going to show you the data. But glycolytic ATP production was much higher in the palmitate-treated cells versus in the non-palmitate treated cells, which again fits in well with this increase in lactate production. So very consistently, we're seeing that palmitate activates glycolysis and activates the production of ATP through the glycolytic pathway, which is not expected and really quite interesting. So why does palmitate activate glycolysis? One reason could be because of this overt change in mitochondrial size and shape. So as I mentioned before, when you add palmitate to the cells, this normally more fused mitochondrial network becomes very fragmented. And we ask, well, maybe this change in mitochondrial shape doesn't change oxidative phosphorylation, but it changes glycolysis. But when we looked at our data, we found that this wasn't the case, because we actually can protect against this fragmentation of mitochondria induced by palmitate if we add glutamine. Then the mitochondrial network preserves its more fused characteristic. On the other hand, the presence of glutamine does not change the glycolytic shift. You still have a lot more production of ATP through the glycolytic pathway induced by palmitate in the presence of glutamine. So there is no direct connection between mitochondrial fragmentation induced by palmitate and the induction of glycolytic pathway. So why is glycolysis induced? Another thing that we had seen before and other authors had seen before is that palmitate induces redox imbalance. And in fact, when we looked at our cells treated with palmitate, we very typically saw a lower ratio of reduced to oxidized NADPH, and also a higher quantity of NADPH, which is very suggestive of chronic oxidative stress, in which you consume NADPH, and that leads to the induction of more production of NADPH. We also consistently saw a small increase in the production of mitochondrial hydrogen peroxide in the palmitate-treated cells. And this production was sensitive to inhibitors of mitochondrial reactive oxygen production, such as sequels, I'll get back to that later, and rodno. So palmitate produces imbalance in mitochondria, and maybe we thought that could be related to this glycolytic shift. So we actually asked the question, is glycolysis sensitive to mitochondrial and cellular redox state? So to answer that question, we first used menadione, which is a compound that generates superoxide within mitochondria. And very nicely, within our hypothesis, menadione induced the production of glycolytic ATP. So that kind of would confirm that glycolysis is induced by reactive oxygen species. The problem with working with reactive things like reactive oxygen species is that they do a lot of different things. And menadione actually pretty much spoiled mitochondria. So we also had increased proton leak in mitochondria, different respiratory rates, higher respiratory rates in these cells. We did not just have the induction of glycolysis. So therefore, this induction of glycolysis and glycolytic ATP production in the presence of menadione could be because of redox signaling, but could also be because there are changes in mitochondrial function. So this wasn't a clean experiment. So we tried a different way to manipulate mitochondrial redox state. And in this case, we used a mitochondrial antioxidant, which can also be a mitochondrial oxidant. All of these antioxidants can also promote the production of reactive oxygen species, which is MitoQ. And this is a quinone, which is bound to a positively charged group So this quinone is targeted to mitochondria. Mitochondria accumulated down their inner membrane potential, and it promotes an antioxidant effect as a quinone within mitochondria. What we found is that MitoQ, despite the fact that it's used very widely in the literature, it's actually in clinical trials for a number of diseases already, has to be used sparingly and carefully, because if you start using higher concentrations of MitoQ, you start having a decrease in mitochondrial function, which is not unexpected. You know, if you have a lot of concentration of something accumulated in mitochondria, you kind of expect that mitochondrial function is going to decrease because of it. So we tried to use more moderate concentrations of MitoQ in terms of this problem with mitochondrial function. And when we use these lower concentrations of MitoQ, we also see a glycolytic shift. So this once again suggests that changes in redox state do change ATP production by glycolysis. But once again, it's not a clean experiment, because this is a pharmacological intervention that is changing the production of ATP by the cell and mitochondrial function. So finally, we tried another system to change mitochondrial production of oxidants, which are Sequel and Sequel. So Sequel and Sequel were developed by Martin Brand's group in small molecule screenings, and they were found in his screenings to change mitochondrial oxidant production from complex 1, that's Sequel, and change mitochondrial oxidant production from complex 3, which is Sequel, and specifically not to change mitochondrial oxidative phosphorylation. Now, once again, that's true in his cells, but when we look at our cells, the effects are a little different, even with very low concentrations. So what we find is that Sequel, the drug that changes the production in complex 3, actually does inhibit respiration in our cells. But Sequel, the drug that inhibits the production of oxidants at complex 1, does not affect respiration. So finally, we have at least one condition in which a modulator of mitochondrial redox state is only modulating redox state and not modulating mitochondrial function and oxidative phosphorylation. Both Sequel and Sequel induced the glycolytic ATP shift. So once again, this shows that changing mitochondrial redox state changes the production of ATP in glycolysis. And I think that's really exciting, because this shows a new mechanism in which glycolysis is regulated, which is a mechanism that is involved in the responses of cells to high lipids. So the effects of Sequel and Sequel are also dependent on the presence of palmitate there, which are the colored bars here. So the conclusions for the lipotoxicity cell part of this talk are that palmitate fatty acids, saturated fatty acids, promote mitochondrial fragmentation, but this is not accompanied by changes in mitochondrial ATP production and oxidative phosphorylation in general. Palmitate also leads, very surprisingly, to enhanced glycolysis, more lactate production, and more glycolytic ATP production. There's a metabolic shift with induction of glycolytic ATP production. Palmitate was known already, and we confirmed this here, promotes redox imbalance. And this redox imbalance is actually the reason why palmitate induces glycolysis. We can modulate glycolysis by changing the redox state of the cell. And we can also modulate the effect of palmitate on glycolysis by changing the redox state of the cell and of mitochondria. So this is quite exciting to me, and I think it should be exciting to metabolism and cancer community, that redox modulation of mitochondria and also saturated fatty acids can regulate glycolysis directly, and that this is a very early metabolic consequence of lipotoxicity. So basically, lipotoxicity and redox imbalance are new modulators of glycolytic function, glycolytic activity. So I have to end by thanking the people who actually did the experiments. As I said, this is basically Pamela Kakimoto's PhD studies. We also had help in this last part of Julian Serna and Vitor Ramos in terms of doing the cell experiments. And of course, help of my whole fantastic group of scientists in the lab. Over the last year and a half, I think I really came to appreciate, I've always thought they were fantastic, but how amazing these people are. Camille Caldeira is our super lab technician. We cannot live without her. Also our postdocs and other graduate students on a whole bunch of other great projects going on in the lab. And finally, funding. We can't do anything without funding. So that's what I had to say today. And let's see if I know how to get out of this presentation now. Stop sharing.
Okay. Thank you so much, Alicia, for the very interesting talk. And we have some questions popping here. So the first one is from Marcos Oliveira. So it's here. So excellent talk, Alicia. Is it possible that oxphos resilience to high-fat diet might change by removing mice from the thermogenic stimuli generated by room temperature?
Thank you, Marcos. You always have great questions. So he already knows this work quite well because he grilled Pamela during her thesis defense. Yeah, it could be temperature dependent, but when we look at the cell model, we also have this resilience of mitochondrial oxidative phosphorylation. So I don't know if it's only that. Of course, in the animal model, the resilience of mitochondria could be because of more turnover. So maybe these mitochondria are being damaged, but they're also being generated de novo again. So there are many reasons. We did not look at changes in temperature yet. Actually, you don't know this, but finally, in the last few weeks, we finally managed to establish thermoneutrality versus cold models in the lab with different temperatures for animals. So hopefully within the next few months, we can have data on different temperatures for animals, which I think is a really important point.
Okay, next question from Joe Fidelito. Hi, Alicia. Do you see the increase of glycolysis when cultured with oleate or palmitate plus oleates?
So oleate does not have these effects. Palmitate plus oleate, I don't think we did. We did look at some effects of palmitate plus oleate in terms of accumulation of lipids and triglycerols in the cells. I don't think we looked at the glycolytic shift, but I may be wrong. You probably should ask Pamela about that. But oleate definitely is not lipotoxic and does not promote that fragmentation of the mitochondrial network.
Okay, let's move to the next one. How glycolysis is activated? A question from Marcos Oliveira again. So how glycolysis is activated by reduced oxidant levels? Is there any evidence that mitochondrial oxidants directly inhibit glycolysis enzymes?
Yeah, so that again is a very good question, and actually it's complicated because both reduced and increased levels of oxidants in mitochondria seem to be activating glycolysis. So it's not a linear activation, which I don't think you would expect in terms of redox signaling anyways. We don't know. We don't know how this is happening. I think a really interesting thing would be to look at the structures of the determinant enzymes in the glycolytic pathway in terms of activity and have a look if they could be redox modulated and actually study that. We haven't done that. So that's a really good point.
Okay, the next one from Amanda Vergueiro. So very good presentation. I'd like to know if you have evaluated autophagy activation in this stressful environment and even the presence of liposomes that may be interfering with metabolism.
Well, I guess you probably mean lipid droplets. So yeah, the question is if these lipid droplets that are induced by lipotoxicity could be changing metabolism. They might actually be protective because they're taking lipids out of the system. So there are lots of questions related to that. And Paula actually has some not published data, but also stuff we don't understand in terms of the accumulation of lipid droplets in this model. So it's something we look forward to understanding. Also, autophagy, Vitor Ramos, one of my students, is specifically studying autophagy in the liver and how that's modulated by mitochondrial function. So hopefully we have data about that in the near future. It's been very difficult to get data for the last year and a half, as all of you know, but I think we do have some really exciting things coming out soon.
So the next one comes from Dr. Gustavo Tibois. So thanks for the brilliant talk, Alicia. Do you think that the alcohol-induced steatosis model would similarly reproduce these effects, or it would be much different?
So that's a good question, because you would expect alcohol to induce oxidative imbalance in mitochondria also. But Anibal Bersesi, my former PhD advisor, actually has data that mitochondria from ethanol-fed animals are more resilient to calcium-induced damage, which involves oxidative stress. And as far as I remember, they also had higher levels of NADPH. So it seems almost the opposite. It seems almost, for mitochondria, ethanol is good. So maybe not so similar. I really don't know.
Okay, so I have a question, and perhaps a last one. So Alicia, and the phenotypes you described for the ANAJ black 6 mice reproduce in humans, similar phenotypes?
So what you would expect is that the human phenotype would be more similar to the animals that have the NNT, because we humans have intact function of this enzyme, and therefore we are capable of producing NADPH efficiently within our livers. And in fact, NASH is not easy to induce with high-fat diets. I mean, all of us have eaten a lot of fat at some point, and most of us will just have benign fatty livers. We will not develop the inflammatory version. So I think that's the big question that we have, which is, what is the difference? What is the change that leads to the inflammatory version? And the idea that nitric oxide synthase was absolutely essential in this came from data with the NNT mutant mice. And when we look at more normal mice, it doesn't seem to be nitric oxide synthase, inducible nitric oxide synthase dependent. So there must be other factors. And the question is this induction of glycolysis important? Because this could change glycerol production and therefore change the production of triacylglycerol. This could change all sorts of different things. And we don't know. This is what we're looking at. We're looking at mechanisms that lead to this disease. That's what we want to know and understand.
Okay, so I'd like to thank you again for the brilliant talk. Thank you so much. And I would like to ask everybody for a few minutes for the next speaker. And thank you so much, Alicia.
Bye.
Bye.
Hello? Could you listen me? Yeah? Bom dia, Alicia. Could you listen me here now? Well, I suppose that yes. Okay. Good morning, everyone. I would like to thank the organizer of this meeting for the opportunity to be here with you all people present, and especially to be here to meet my good friend, my friend for 14 years, maybe, Hervé Chneiweiss, that go talk with you. And Hervé is a very good friend, 40 or 45 years, I don't remember, a long time ago. And Hervé is a medical doctor, PhD, is a neurologist and a neuroscientist, research director of the CNRS, the Centro Nacional de Recherche Científica. He has been involved in neurogenetic research on diseases such as cerebellar ataxias, and then the molecular mechanisms involved in glioplasticity and the development of the brain. Technical approaches include proteomics, metabolism, epigenetics, cell cultures, animal models, single cell. He has published over 170 original scientific papers. He is currently director of the Neuroscience Paris Seine, IBPS Research Center, CNRS and Inserm, this Institut de Santé et Recherche Médicale, Sorbonne University. He is also involved in bioethics. First, 2000-2002, advisor for life science and bioethics to the Minister of the Research and Technology, member of the Scientific Council of the French Parliamentary Office for Scientific and Techniques Assessment 2003-2016, member of the National Ethics Committee 2017, and currently is chairman of the Inserm Ethics Committee and of the UNESCO International Bioethics Committee. He is also an expert in ethics of neurotechnology for OECD, and was a member of the WHO Committee on Human Genomic Governance, former editor and chief of Medicine Science, 2006-2016. He has published several books for the general public. The last test is Notre Cerveau, l'iconoclastre, de 2019. This is a profile resume of this young researcher that I have a very good pleasure to be here in front of here by virtual conditions, virtual situations. It is at the moment that to talk about the colleagues, your talk, please.
Thank you so much. Thank you so much for the organizer to invite me. Thank you so much to you, Vivaldo, once again for so kind words that you said. I'm going to try to make my presentation. That's it. And as you mentioned, Vivaldo, we met in Paris so long time ago, but like that we are still young forever. And what I'm going to present to you today is a work that indeed we initiated together, Vivaldo, in Paris and in Rio. And many people that are in the audience, most likely at UFRG, or that are now assistant professor or professor in UFRG, and in other universities in Brazil, participated to the settlement of this work. So what we are going to speak about today is the most aggressive brain tumor, namely glioblastomas. Among brain tumor, in the last WHO classification, you have various names and grade. It's not because the tumor is named astrocytoma that the origin of the tumor is astrocyte. You have various origins, and in the team, we demonstrated that it could be astrocyte by a reprogrammation toward undifferentiated stem-like cells. But it could turn also toward oligodendroglioma, which is just means that it looks like an astrocyte or it looks like an oligodendroglioma. But what we are going to speak about is the most frequent one and also the most aggressive, as you can see. As you can see from the 5-year survival, the glioblastoma. And for a long time, it was there were discussions to try to make classifications. The only real classification we can have is this mutation in isocitrate dehydrogenase enzyme, EDH, either EDH1 or EDH2. And just to follow on the previous talk, EDH1 is mitochondrial enzyme. And in the two diffuse gliomas, oligo and astrocytoma, you have this IDH1 mutation, not in glioblastoma in most cases and in the terminal phases. And you have some elements of better prognosis in oligodendroglioma, this deletion in 1p19q chromosome. As you can see, the incidence of glioblastoma is not that much. It's 5 out of 100,000 per people per year, and the similar repartition all over the world. It's mainly a tumor of the aged subject, but if it's not big in incidence, it's terrible in death because of this very short-term survival. So as I already said, we had many attempts during the last 10 years or so to make some kind of stratification or molecular stratification of brain tumors at large or glioblastoma particularly. But it's really not. Yes.
I'm sorry to interrupt you, but I think your presentation is stuck on the title slide. Maybe you should come back to the presentation, and it's only in the first one.
So you think that there is a problem with?
Yeah, you can share it again. Sorry, technology doesn't work as we want them to work sometimes.
What slide are you looking to now?
It's still in the presentation. In the first one is the title slide.
So it's not moving?
No.
Strange.
Maybe you should try to share your whole screen.
So now you see
Yeah, now we can see it, but you should put it in the presentation mode because we can see all your PowerPoint.
Presentation mode, so it's... What do you see now? Is it moving?
No, try to move it for the next one.
I moved to the next one.
Yeah, no, it's not moving.
Ah.
Oh, okay. Yeah. Now it is. Now it is moving. Okay, so it's a little bit slow.
So now you are on the...
Glioblastomas are highly heterogeneous tumors. This is the slide that we're seeing now.
Oh, so
Okay.
okay. I'm going to try to see. Maybe I've no. Sorry for this. Do you see something?
We're seeing that slide from a.
Okay, so I'm going to comment, and if there is any additional problem, just let me know, Luis.
Okay, thank you.
So glioblastoma are highly heterogeneous tumor, and you have just here an illustration of this intratumoral heterogeneity. As you can see, if you make several types of biopsies, you can have the down score of the tumor. You can have it's highly vascularized. So you see this number of vessels. You can see also era of necrosis. And obviously, at the limits of the tumor, you see an infiltration of the normal parenchyma by the tumor. So considering this heterogeneity, you are going to have also an heterogeneity when you are going to make the transcriptome or the genetic analysis, including the number of chromosomes. And Marie-Pierre Génier tomorrow will speak about our attempt to look to this heterogeneity at the single cell level. I'm not going to speak of that today. Do you see the next slide? So I'm going to make as if you see the next slide.
Unfortunately, I think that it's still stuck. It's still on glioblastomas are highly heterogeneous tumors. Maybe if you press the bottom lecture in the top right of your screen, where there's a play, like a play icon.
Yes. Do you see something now?
Now we are seeing microenvironment factors modulate.
Okay. So it was just to say that this heterogeneity is clearly related to some microenvironment. And our team contributed to the analysis of this microenvironment in collaboration with Julie Gavard. We were able to look to the interactions between endothelial cells and a particular population of glioblastoma cells that are the glioblastoma stem cells. Glioblastoma stem cells are glioblastoma cells endowed with stem-like properties and with tumorigenic properties. And this dialogue include molecules secreted by the endothelium, like SDF1 or TGF-beta, that are activating some signaling cascade inside glioblastoma stem cells, allowing them to survive, to self-renew, and to make some pseudo-pericyte. When you look to a glioblastoma, you see around the neovessels some tumoral pericytes. Some molecules that are secreted by glioblastoma stem cells, such as HDGF or semaphorin 3A, call for new endothelial cells and some pericyte to create these new vessels and to build up these new vessels, in addition with other known factors such as VEGF. And this results in endothelial cell migration, blood vessel formation, and change in blood vessel permeability, which explain why on MRI you see the gadolinium, the contrast product, moving out the blood to enhance the contrast of the tumor. So recent studies, I should say during the last 5 years, clearly illustrated that considering this intratumoral heterogeneity, the various subgroups that were once described, such as proneural, classical neural or mesenchymal profile of glioblastoma. We're not.
I'm so sorry again, but it's still stuck. Maybe you should try to stop sharing your screen and try to make it again, but this time you should try to share your whole screen, because I think it's you're sharing only your PowerPoint. So you should say like PowerPoint presentation.
I share screen, and now I say share screen. Okay.
And then you click like full screen or something.
Okay, full screen. Okay. And now I try again. You see something?
It's not showing up yet, but maybe there is a delay.
On my screen, it's the slide now.
Okay, here we don't see the sharing.
Okay.
Sorry. Can you go back and in the StreamYard studio there is like this icon written like share or something, like there is a computer screen and a plus sign on it. Okay, now I see something here. Yes, so put your PowerPoint presentation. Now I think it's going to work. Great. Now it is good. I think it's going to work. Sorry, I'm so sorry.
No, I am really sorry that that it's not working, because it might be difficult to follow up the presentation.
Yeah. Okay, I'll leave you. Thank you. Thank you.
No, don't, Gustavo, don't. It's just fun.
Yeah.
Okay.
Okay.
So I just get back to the fact that what was presented at a given time, like several profile of glioblastoma, such as proneural, neural, or mesenchymal, are indeed just the dominance of cells with such profile according to the place they have in the tumor, more or less around a vessel, more or less perinecrotic. And within a given tumor, you can find all the types. And you can find functionally divergent cell population that have stem properties, that are proliferate or not, that migrate or not, that are call for vessels or not, that are tumorigenic or not, and so on and so forth. I hope you see the next slide, which try to illustrate that this is highly, it's also evolutive during time and dependent on the various interactions of cells, for example, interaction of tumoral cells with myeloid cells, but also according to treatments. And it was shown that radiotherapy, for example, is moving cells from a proneural phenotype toward a more mesenchymal phenotype. So our team, a long time ago now, because it's more than 10 years, used a very simple paradigm in collaboration with Thierry Virol in Nice to try to investigate what is happening in the glioma stem cells according to the change in their microenvironment. And we use a very simple thing. We expose the cell for 24 hours to serum, and it was enough to observe a decrease in their tumorigenic capacities. So we tried to investigate for the molecular mechanisms that were underlying this loss of tumorigenicity, and more specifically, what types of microRNA could be involved in that, and making a differential analysis between cells exposed with tumorigenic properties and cells having lost their tumorigenic property. It was possible to evidence a certain number of differentially expressed microRNAs, and particularly a group of 5 microRNA 302 and 3767, which is a cluster of 5 microRNAs. And we wondered if it was enough to express this microRNA to explain the different properties, and indeed it was the case. To make a long story short, when you take cells that are expressing these clusters of 5 microRNA, as you can see with this illustration of xenograft in nude mice, here the naive cells are making a big tumor, and the cells that are expressing the cluster of microRNA are not. Is it relevant to what is happening in patients? And the answer is yes. As you can see, according to ERAS, with various densities of tumoral cells, you can see lower or higher expression of some of these microRNA. And the more you see the subpopulations that are expressing more of these microRNA, you have a lower score of proliferation. So as you will see, we use this paradigm of cells expressing or not microRNA was used in following studies, and in a series of studies, once again with a team of Thierry Virol, we explored the mechanisms underlying this effect, and I'm not going to describe these studies because they are already published for a long time, but we dissected molecular mechanisms expressing the effects of microRNA 302, 3767, including receptors such as CXCR4 and their signaling pathway in the cells, and also a balance between the factors DOC4 and beta-catenin with the influence of GSK3 beta to control the proliferation and the differentiation of glioblastoma cells. On the reverse way, we were also able to demonstrate that when you remove serum and you put back your glioblastoma cells in a defined medium for stem cells, they are going to recover their stem and stem-like and tumorigenic properties. And other microRNAs are involved in the recovery of these stem and tumorigenic properties, including microRNA 18A that we described. And we also dissected the molecular mechanisms underlying the effect of this microRNA. And these mechanisms include ERK, include the expression of other cascades of microRNA, such as this one, 199-3P, and factor and transcription factor EGR1. So as you see, the phenotype and the properties of glioma cells are not fixed. And it's not a hierarchy of cells, which like in the normal stem cells go from undifferentiated toward the differentiated cells with a terminal differentiation. In glioblastomas, and most likely in many cancer, solid cancer at least, you have oscillations between stem and tumorigenic state and more differentiated or pseudo-differentiated status. And we were wondering if these oscillations were not at the basis of cellular heterogeneity, and if these oscillations were not producing the various cell types that we are observing in the tumor. And we focused more our attention on metabolic pathways coupled to epigenetic regulation as drivers of cell diversifications. And I will show you also some works on transcription factors. So why working on metabolism? I think that you have plenty of reasons. But one reason is also that metabolic reprogramming is known for a very long time, but was seen for this very long time as the support of cell functioning. For example, when cells are migrating, it's clear that they need energy to colonize some surrounding tissue. And because they need this energy, it's normal to see change in glycolysis. It's the famous Warburg effect, and oxygen availability, because when the tumor grows, you have areas of hypoxia. We had another hypothesis, wondering if, because of the oscillations, because of the change in expressions of a certain number of factors, including metabolic factors, these metabolic reprogramming were not, in some cases, driving the change and also driving the oscillations. So we took again our paradigm of tumorigenic and non-tumorigenic according to the expression of microRNA 302, 367, and we made a metabolic profiling to compare the two. It was possible in these conditions using gas spectrometry to see various increase and decrease in metabolites, whether it was intracellular metabolites or extracellular secreted metabolites. And you see just the number here. And we focused our attention in a work that Gustavo knows very well on the catabolism of GABA, because you see all these numerous metabolites that are in green decreased, but in red increased. And we were wondering why GHB was increased, because you can easily think of various factors that are in conjunction and in flux decreased, but why just one was increased in this condition? And you can see that the increase in cells expressing MIR was true in the cells as well as in the secretome. So to make a long story short, making some bioinformatic analysis on the targets of microRNA 302, it was possible to make the hypothesis that the target was a gene named ELDH5A1, producing an enzyme named SSADH for succinic semi-aldehyde dehydrogenase. And as you can see here, indeed, in cells expressing MIR302, you have a strong decrease in expression of ELDH5A1. Trying to investigate this with silencing RNA, acRNA against ELDH5A1, it was possible to see a decrease in the expression of SSADH in glioblastoma cells, but also very interestingly in other glioma stem cells we were able to derive from pediatric tumors, deep infiltrating pontine tumors. You can see that when you knock down this gene, you knock down clearly the expression of the enzyme, and in parallel, you observe an increase in GHB levels. So this is strongly supportive of the targeting of ELDH5A1 by microRNAs. Once again, is it relevant for patients? So we return to our tumors and to the histopathological analysis of our tumors. And it was possible to see micro-eras, micro-populations, where overexpression of SSADH was associated with high proliferation, as seen here with Ki67, and undifferentiated tissue, whereas when in eras of low expression of SSADH, you have also more differentiation and a low proliferation. And it was possible to make an index of GHB, the increased product in glioblastoma cells, to alpha-ketoglutarate. You will see later why. We were wondering what was the effect, the biological effect of this, and clearly the overexpression, the exposure of cells to GHB itself trigger a decrease, an inhibition of cell proliferation. You can see here on a part of our collections of glioblastoma stem cells that it was possible to see inhibition on all the cells that we treated, whether it was adult glioblastoma stem cells or high-grade glioma stem cells from pediatric tumors. We see also that when we knock down ELDH5A1 and have a decrease in SSADH, we also inhibit proliferation. And here another representation by incorporation of BRDU, you see that the S phase, so the proliferation phase, is completely inhibited either by exposure to GHB or by the knockdown of ELDH5A1. Next step was to look for differentiation of cells, because as I told you, GSC are defined by their stemness factor, and it was possible to evidence that GHB was taking the cells out of stemness. Here you can see from the clonality assay that exposure to GHB makes the cells losing their clonality very rapidly. You can see that exposure to GHB is also triggering differentiation of cells with loss of OLIC2, loss of NANOG, and on the contrary, an increase of beta-tubulin or GLT1, which stand here as a pseudo-differentiation mark of the cells. And finally, when we tested the cells in vivo, in xenograft, it was possible to see that GHB was inhibiting tumorigenicity. As you can see here, when you knock down LDH5A1 with shRNA in this case, you strongly decrease the size of the tumor and the number of tumoral cells that you can recover from a xenograft. What is the mechanism of action of GHB? For this, we made some kind of computer modelization about taking as basis the structure of GHB and in comparison alpha-ketoglutarate. And we tested if GHB could be a competitor of alpha-ketoglutarate on a certain number of alpha-ketoglutarate-dependent enzymes. And it turned to be the case, at least for this enzyme, TET2. This enzyme is involved in the transition from 5-methylcytosine to 5-hydroxymethylcytosine. And you have to know that 5-hydroxymethylcytosine was often named the fifth base of the genome because it's signal genes that are ready to be expressed very usually. And you can see here that exposure of cells to GHB strongly decrease the level of 5-hydroxymethylcytosine without really changing the level of 5-methylcytosine. And looking more closely to TET2, it was possible first to modelize the docking of GHB at the same place as the docking of alpha-ketoglutarate, to show that when you overexpress in a cell TET2 and then expose to the cells to GHB, you have a strong decrease in 5-hydroxymethylcytosine. And finally, to show that when you knock down TET2, you are able to see the decrease in 5-hydroxymethylcytosine and the effect on cell proliferation and cell self-renewal. So to make the summary of this, and to thank Luis Gustavo when he was in our team in Paris a long time ago, a work that was then completed by Elias Elab. When you have the glioblastoma stem cells, you have a very high catabolism of GABA to fuel the TCR cycle through SSADH. And in this case, TET2 is very active, producing high level of 5-hydroxymethylcytosine. When you expose the cell to GHB, or when you knock down SSADH, if you knock down SSADH, you produce more GHB, you inhibit TET2, and you inhibit 5-hydroxymethylcytosine. The more interesting thing is that in these cases, whether it is GHB, whether it is the knockdown of SSADH or the knockdown of TET2, you inhibit tumor growth. And now we are searching for some molecules that could do the same. Interestingly, for example, valproate is known to inhibit SSADH, and Elias Elab was able to demonstrate that exposure of cells to valproic acid was able to increase GHB and decrease cell proliferation. Unluckily, clinical trials in human using valproic acid were not successful at this time to improve as neoadjuvant the treatment of glioblastoma, but this could be because the concentrations reaching the tumor is not enough. Just to illustrate that we have other targets also to study, just here unpublished results on the methionine metabolism reprogramming. We observed an increase in methionine and in downsized products such as nicotinamide in our cells, and a decrease in SAH, S-adenosyl homocysteine, and in one MNA. Considering the fact that we previously published the fact that NNMT was overexpressed in glioblastoma, we were interested in looking more closely to methionine metabolites. And it was possible to show that these methionine metabolites, at least pretreatments of the cells in vitro with these methionine metabolites, were able to strongly decrease proliferation, as illustrated here, a work by Ashwin Narayan and Merka Sorti, now close to be submitted for publication. And just to give you the final result of these metabolites, but you will have the whole story soon. Here again, these cells that were exposed to this metabolite as a pretreatment had a delay, in not a complete inhibition, but at least a delay in cell engrafting, and this resulted in increased lifespan of the mice after this xenograft. So you see that we have plenty of modules to explore, and this open very interestingly a new aspect. And a very surprising and interesting aspect looking for these metabolites was to observe that at the end it was changing an histone mark, and in this case it was an increase in H3K27 3 methylated marks. And you know that these marks are repressive marks. This led us to go further and make a more systematic analysis of these marks, looking for active marks, K3, or repressive marks, still in our model of tumorigenic or not. And this allowed us to evidence a lot of transcription factor, but more particularly study this one, RNT2, because it appears at the core of a dense network of these transcription factors. And also because clearly you see here that RNT2 is downregulated in cells having lost their tumorigenic properties. It was clear that marks of active expression were highly expressed in highly tumorigenic cells, whereas repressive marks were expressed for RNT2 is cells having lost their tumorigenic properties. It was possible, once again, to check in the real tumors, in patients' datasets, that RNT2 was expressed in highly proliferative cells and poorly expressed in low proliferative cells. And clearly, using various data cells and various bioinformatic tools, it was possible to see that RNT2 is functionally associated with a molecular signature of tumorigenicity, a signature of aggressiveness. Very interestingly, it was possible also to show that RNT2 was a master gene controlling some other factor, including other transcription factors such as OLIG2, PU3F2, or SOX9. To return to the in vivo system, we were able to show that RNT2 drives glioblastoma cell aggressiveness. When you knock down RNT2, you decrease tumorigenicity, you increase the delay for cells to engraft and produce a tumor, and you increase the survival of the mice. With some very aggressive tumor, it's just an increase in survival. In some other cases, you see that the survival is impressive. So it might be also dependent on the profile, the genetic profile. So to thank this brilliant postdoc we had, doc and postdoc for Alexandra and postdoc for Elias, and to make a summary, RNT2 is a master gene controlling a cascade of other factors and controlling the tumorigenicity of the cells. And very interestingly, to feed back to this metabolic story, you can see that among the pathways and among the modules that RNT2 is controlling, you have a block of metabolic pathways, essentially in the lipid cellular pathway, cholesterol, long-chain fatty acids, but also glucose catabolites and sterol. And it will be very interesting to go further on what kind of metabolic pathway are controlled by the expression of RNT2, a story which is now ongoing in our lab. So to make the summary of what I presented to you, and just to before what Marie-Pierre will present to you tomorrow. You have clearly a control of this exceptional plasticity of the glioblastoma cells by metabolic modules and by epigenetic modules. And it's not two different modules. Metabolic reprogramming target epigenetic regulation, and epigenetic regulations in turn once modified the metabolic modules. I presented to you that some microRNAs such as 302, 367 are able to control metabolic modules, GABA catabolism, methionine metabolism. It could result in modification of DNA, such as 5-hydroxy methylations with STAT2, but also in change in histone marks, changing from active to repressive for the methionine metabolism, but also for ARNT2. And these modules that are controlling the fate of the cells could be now targeted, and I think that it will be one of the aspects of the next presentation. I'd like to thank the different members of the team that are listed here, and I tried to thank them along the presentation. The present member, but also our collaborators. And among these collaborators, it's wonderful again to be back with all of you at UFRG, including obviously my old friend Vivaldo and Gustavo. Thank you for your attention, and sorry for the technical difficulties at the beginning of the presentation.
Thank you, Hervé. Thank you very much. Merci beaucoup. As you may have seen, Hervé was one of the supervisors of my doctoral works, and I'm very happy to be here with you today. And we are happy also for you to have accepted our invitation. So I am a little bit biased to make questions for the first part of your work, but I have a first question about the ARNT2 protein. It is very implicated with hypoxic conditions, right? And you have showed that the expression of ARNT2 is very important during the differentiation of the glioma stem cells, right? So do you think that in hypoxic conditions, how this response would change the metabolism of those cells? Do you think that it would be like posing some differentiation too?
Well, do you see my slide?
Yes.
Yes. So yes, ARNT2 belongs to the family of BHL of transcription factor, where you have also hypoxia-induced factors. And these factors, you have two classes of these factors, class 1 and class 2, and you need one factor of the class 1, such as ARNT2 or ARNT, to interact with another factors of the class 2 to make the dimer and make something working. So ARNT factors were considered as a factor of hypoxia. As you can see in this diagram, and we are still working to try to evidence which of these factors is the real partner of ARNT2, but as you can already see from these factors, there is no clear correlation between the expression of ARNT2 and one of the HEF factors that was involved in glioblastoma development and in hypoxia. And particularly this one, HEF2A, was reported to be important in glioblastoma, and you see that there is absolutely no correlation between ARNT2 and HEF2A. So two points. One, we think, we don't have a clear demonstration yet, but we think that ARNT2 is not directly related to hypoxia. It could be in certain cases, but Most likely, most of the time, it's not related to hypoxia, and that these factors that are strongly regulated by hypoxia are also involved in other ways of regulating. And for example, you can see here that HEF2A is densely expressed in perinecrotic zone. So it could be hypoxia, it could be also the acidification of the pH. And you can see that in this era, it's ARNT, which is highly expressed. So most likely the dimer is between ARNT and HEF2A. So I hope I answer partly to your question, but at that time, it's the best answer I can do.
Great, thank you very much. Yes, it's because hypoxia is so involved in regulating metabolism, and also this idea of metabolites regulating differentiation in cancer cells is very interesting and very intriguing, right?
Yes, but as was already said by Alicia before, you always have to think about metabolism, and hypoxia is just one aspect or one stress that cell have to respond to. You always have to think metabolism as fluxes, and so it's always a question of balance of what is getting in and what is getting out, and always a matter of fluxes. So it's clear that hypoxia is going to impact the cell as a stress, but it's not possible to say that all the cells in a tumor are stressed by hypoxia. A few number of them are so, but you need to remember that most of the stem cell niche in the glioblastoma are in highly vascularized era. So they are in contact with vessels or close to vessels. Obviously, these vessels are not with the same permeability as normal vessels. Very often you see vessels with a very low flux of blood, but you cannot say that all the cells in a glioblastoma are hypoxic. And specifically the cells with high proliferation that are often far from hypoxia zone. And these ones are highly expressing RNT2. So most likely we have to go further and to discover more than simply hypoxia.
Great. We have some questions here from the audience. So Gabriela is saying, thank you for the talk. Concerning the first part of your talk, you mentioned that clinical trials failed probably due to the fact that the inhibitors could not reach the tumor. So it continues. Is there any correlation between poor responses to this inhibitor and the status of brain-blood barrier intact or not in glioblastoma patients?
This is one of the big challenge of any treatment of glioblastoma. On one side, it should be easy to reach the glioblastoma cells because, as previously said, the blood-brain barrier is disrupted because of the high permeability of the neovessels, disrupted because of some era of bleeding or necrosis. So normally the molecules that are in the blood should reach these glioblastoma cells. In fact, they are not, for many reasons. One is that this hypervascularization is strangely associated with a strong perturbation in the blood flux. So it's not clear which cells are reached or not. It's not also because what I illustrated very early in my presentation, and maybe I can return to one of the first slides. You have a dense part of the cells that are in infiltrative zone, here in the lower panel. And in this case, the blood-brain barrier is maintained. And so you have cells either proliferating or dormant cells that are going to stay in this infiltrative part. And we need to find molecules or techniques to reach these cells as well.
Great. And do you think that you could like develop another kind of therapy for enhancing GHB, like genetic approaches, like decreasing the ALDH5A1?
Well, we have yes, we have several ways that we try to explore. One was to combine various molecules and making some repurposing. The strategy that was so great with Susanna Sadkan to demonstrate that prazosin was a good anti-tumoral molecule. So Elias tried to do that with valproate. Another way is to try to bring the molecule as close as possible to the cells. So using some wafer, for example, as it was done for carmustine with the gliadel wafer. So you have some strategies to create some biomaterials that should bring directly the molecules in contact with the cells. Well, it's still ongoing work.
Great. Thank you very much. Thank you very much, Hervé, for showing up and showing your input on your work, on your excellent work. And so we are gonna have our last speaker in a few minutes. We are gonna go for a brief break, and we will come back soon, okay? Thank you very much, Hervé. Bye.
Thank you. Have a nice day.
Yeah. Okay, hello everybody. I hope you're hearing me. So for the last part of our first day of this meeting, I am very glad and very happy to introduce my colleague that was one of the supervisors of my postdoctoral training. And please welcome to Professor Dr. Richard Possemato. Thank you very much, Richard, for joining us today. And well, Richard is an associate professor in the Department of Pathology at the New York University School of Medicine. And for his doctoral work, Richard studied cellular models of human cell transformation under the advisement of Dr. William Hann at the Harvard Medical School. I hope I get this name right. And his postdoctoral studies with Dr. David Sabatini at the Whitehead Institute, Richard developed genetic tools and designed culture systems to investigate the contributions of the nutrient environment on cancer cell metabolism. And he described a key role for the serine biosynthesis pathway in human breast cancer. Richard is a professor at the pathology department at the University of New York since 2014. And he's establishing a research program that focuses on understanding altered cellular metabolism in human disease with an emphasis on iron metabolism and breast cancer models. So, Richard, thank you very much for joining us today. And I'm really happy to see you despite this, this moment of social distancing. And I hope that we will be meeting in person soon, right?
Yeah, thank you so much. It's good to see you as well. I wish I could see everybody who's here, but thank you for joining me in my office in New York. I'm gonna share some data if I can do that. Let's see how this goes. Okay, tell me if you can't see this because I don't have any feedback.
Yes, yes, we can see it.
Okay, great, thank you. Great. So this talk today is about a journey that I've taken since my postdoctoral work in Boston, where I was working on cellular metabolism in cancer and got interested in iron and in iron-dependent processes. And I'll tell you a little bit about my journey there and some of the recent work that we've done to understand iron in cancer. And I'll just jump into it. So this project started with this idea that oxygen is very different in a lot of the settings that we care about. Atmospheric oxygen, oxygen in tissues in our body, hypoxic regions of tissues. And you could think of, sort of the cells that you might grow in a cell culture dish as being somewhere towards the right end of the spectrum, whereas a tumor, which is a system that we care about, would be more towards the left-hand side of the spectrum. And of course, we use one as a model for the other. Most of the tissues in our body are somewhere in this intermediate range. And so, how does this affect metabolism and how does this affect the requirement for certain metabolic genes? And so we had done a screen in these conditions and we found something that I was actually hoping to find something that would kill cancer cells in tumors, but we found quite the opposite. And this really interesting phenomenon that I'm showing you on the left, which was that when we suppressed this gene, NFS1, and I had no idea what this was at the time, or what an iron sulfur cluster was at the time, but when we suppressed this gene, we saw this really nice suppression of cell growth in cell culture that didn't happen when we shifted cells to a low oxygen concentration. And NFS1 is a cysteine desulfurase, so it takes an atom of sulfur out of the amino acid cysteine and donates it to this cofactor called an iron sulfur cluster. And this happens in the mitochondria, and then these cofactors are chaperoned either out of the mitochondria for their use in a variety of cytoplasmic or nuclear proteins, or they're incorporated into a variety of mitochondrial proteins. And interestingly, based on what I've told you, you might not be surprised that when we suppress this gene in a KRAS P53 mutant lung cancer cell line that we got from a colleague of mine, that we don't see much difference in growth in the hypoxic tumor microenvironment. However, when we did this in by directly instilling cells into the lungs of the mice, we now see a re-requirement for this pathway, suggesting that within an organism, the oxygen concentration that a cell might experience can be different, and that can select for the requirement of a metabolic pathway. And this is just IVIS images that were quantified in this middle panel here. And we spent a lot of time trying to figure out why this was the case. And my initial reaction was that oxygen was somehow damaging the cell in a way that involved ROS. But really, we could only rescue this phenotype of suppressing iron sulfur cluster synthesis by placing cells in a low oxygen environment. Nothing that we did with respect to giving any kind of antioxidant. We tried to rescue this with everything we could think of. Only shifting cells to low oxygen would give us a really nice rescue of cell viability. And so it was oxygen itself that was really important for maintaining the viability of these cells, where we had low levels of biosynthesis of these cofactors. And one of my favorite pieces of data that a student of mine collected was doing this assay, this in-gel assay for the enzyme aconitase, which is an iron sulfur cluster dependent enzyme. It's a member of the TCA cycle. There's also a cytosolic form. And so you can see these two forms, they resolve well on a gel. And so the purple color is qualitatively linked to the activity of the enzyme. And so you can see that the mitochondrial and cytoplasmic enzyme have some activity. At 3% oxygen, this is slightly affected. And it seems like the cytosolic form is more effective, which is kind of interesting and may be a reflection of the differences in oxygen concentration in those two subcellular compartments. That's one possible explanation for this. But then we shifted these cells into atmospheric oxygen concentrations. And you can see two things. First, that in general, you reduce the activity of this enzyme that is dependent on this iron sulfur cluster cofactor. And when you suppress the biosynthesis of the cofactor, now you really are starting to lose enzyme activity both in the mitochondria and in the cytosol, suggesting that you're going to start to see frank dysfunction of iron sulfur cluster containing proteins in those compartments. And so our assumption, or our model based on these data, is that NFS1 is making these iron sulfur clusters. Then there's some war between biosynthesis and oxidative damage of the cofactor, and that you need these cofactors in some resident protein to do some cell essential function. And when you block this with RNAi, the oxidative damage wins out over the biosynthesis, and you lose the cell essential function. And so, so what is this? What are these blue ovals? Another phenomenon that we noticed that we thought initially was related was that the cells, when suppressed, were really sensitive to oxidative damage. And this is a bit in contrast to what I just told you, that that oxidative stress could, that iron chelators, that antioxidants couldn't rescue this effect, but oxidants could make it much, much worse. And so you can see here with this glutathione synthesis inhibitor, but really any kind of oxidative stress would lead the cells not just to stop growing, which is what happened when you put them in high oxygen, but to basically lyse. And this is what the cells looked like over here on the right. These cells here were really just exploding. They were lysing. And we came to realize that this was a type of cell death that's been described a lot recently called ferroptosis. And I'll describe a little bit about why this happens. But first, I need to talk a bit about how iron is managed in the cell. And so there is this pool of iron in the cell that's called the labile iron pool, from which the iron sulfur cluster synthesis machinery can draw to make the cofactor. And then can be loaded into these resident proteins. If iron sulfur cluster synthesis is inhibited, or if iron is lost, this aconitase enzyme, which I mentioned, can undertake a different conformation and a different function, such that it can bind to these hairpin structures in certain mRNAs, either in the 5' or 3' end. And these are called iron responsive elements. And this protein is now called an iron responsive element binding protein. And binding of IRPs to iron responsive elements will either inhibit translation or stabilize a message. And so in a situation where there's low iron, this will get activated, and you'll inhibit translation of proteins that store and use iron so that iron becomes available. And you'll stabilize the message of proteins that bring iron into the cell. And so you bring more iron into the cell by stabilizing these messages. There's another arm of this pathway here on the right that at the outset of our work was thought to operate. Thus, basically that iron would bind to this E3 ligase FBXL5, that would then lead to the degradation of IRP2 in an iron-dependent manner, such that when iron is present, this would get degraded. When iron is absent, it would get stabilized, and it would similarly activate this response. And so you can look in the conditions that we were using at this protein transferrin receptor, which is stabilized when you inhibit iron sulfur cluster synthesis, and this protein ferritin heavy chain that's suppressed when you inhibit iron sulfur cluster synthesis. And these two effects are indicative of activating this IRP-mediated iron starvation response. And this happens at high oxygen, and this happens also at low oxygen, which is interesting because a lot of the experiments that we are doing to make these cells where we suppress NFS1 at low oxygen are perfectly happy to grow, but they still activate this iron starvation response, suggesting that this is a very sensitive, an event that's very sensitive to iron sulfur cluster deprivation. Okay, so this is a summary of what I told you. So iron can come in. Here we have cysteine coming into the cell. It can be used for an iron sulfur cluster or to make glutathione. Iron sulfur clusters are used in these IRP sensors. When the iron sulfur clusters are lost, you trigger this iron starvation response, you increase the labile iron pool in the cell, and then this can cooperate with any kind of oxidative damage to lead to ferroptosis. And so the situation that you get when you suppress iron sulfur cluster synthesis is really one where you've overloaded the cells with iron, and then any other kind of oxidative stress can induce damage to cellular membranes because iron is such an oxidative molecule. It needs to be protected in these ferritin cages. If the labile iron pool is too high, then you can now get oxidative damage and cell death. Okay. So I just wanted to share a few interesting tidbits about iron sulfur clusters and human disease and things that we've been thinking about and others have been thinking about that are fairly interesting. So in SF3B1 mutant MDS, there are hypomorphic mutations, or in hypomorphic mutations in the germline of ABCB7, you can get inhibition of this particular protein. And so SF3B1 mutant MDS missplices ABCB7, so you have low levels of this protein. And then in this disease, Friedreich's ataxia, you could have triplet repeat expansion of this other protein, frataxin, and so low frataxin levels. And so either of these changes, low expression of ABCB7 or low expression of frataxin, will hinder iron sulfur cluster synthesis and activate this iron starvation response, leading to an increase in free iron, and in the case of erythroblasts, a decrease in heme. And so all of these, or both of these conditions, SF3B1 mutant MDS and germline mutations in frataxin, yield phenotypes that are indicative of oxidative stress in the case of frataxin or ABCB7 mutations leading to neuronal cell death, or in the case of SF3B1 mutant myelodysplastic syndrome, leading to a blockade in erythroblast differentiation into erythrocytes, and therefore sideroblastic anemia. And so these downstream effects of mutations that are observed either in cancer or in human diseases impact iron sulfur cluster synthesis and may be responsible for some of the pathology for these diseases through constitutive activation of these IRPs. And so these are hypotheses that are currently being tested by us and others, and I think are attractive for explaining some of these disease pathologies. Okay, so I mentioned a bit about how inhibiting iron sulfur cluster synthesis in one breast cancer cell line can induce the iron starvation response and sensitivity to ferroptosis. Since then, we've expanded this to other breast cancer cell lines, and we can see an interesting correlation between cell lines that are activating the iron starvation response by increasing the transferrin receptor or decreasing ferritin, and the cooperation with oxidative damage, so in this case, arrestin. So some cell lines do a very good job of activating the iron starvation response, and some don't when we inhibit NFS1. And those that do show synergy with oxidative damage, and those that don't do not, consistent with what I just told you. And understanding why there's this dichotomy is interesting and something that hopefully we can tell you about someday. We can tell you, though, is that this effect is dependent on the transferrin receptor. So if you block the ability of the cell to activate the transferrin receptor when you inhibit iron sulfur cluster synthesis, you also block their ability to undergo ferroptosis, and therefore import of iron is sufficient to induce this phenotype, is necessary to induce this phenotype. Okay, so knowing that, we wanted to ask a little bit more about whether IRP1 or IRP2 was responsible for this effect. And based on the pathway architecture that I just showed you, we would expect that IRP1 would be solely responsible for transmitting this signal of iron sulfur cluster deficiency to the iron starvation response. And so so here's the cells that have both IRP1 and IRP2, and we deleted IRP1 in these cells and actually several cell lines, and we were surprised to find that, in fact, the activation of the iron starvation response was really unchanged, suggesting that iron sulfur clusters really could be signaling in some unknown way through this other iron responsive protein. And when we knocked out IRP2, we in fact saw the same phenotype that in fact this side of the pathway can operate as well. And so it seemed to be the case that both mechanisms were operating. And deleting either IRP1 or IRP2 didn't affect this ability of these cells to undergo ferroptosis when we damaged the cells with some kind of oxidative stress. So you really needed both IRP1 and IRP2, or either IRP1 or IRP2 to signal through this pathway. Okay, and then if you deleted both of them, you no longer got activation of the iron starvation response, and you rescued this sensitivity to ferroptosis. So these cells basically became completely insensitive to any kind of ferroptosis inducing agent without this iron starvation response. Okay, I'm going to skip ahead a little bit to the next question, which I think is also fairly interesting, which is to understand what these blue ovals are downstream of NFS1 that's responsible for these cell essential functions when you inhibit iron sulfur cluster synthesis. Some things that can also do this are perturbed lysosomal pH, which blocks iron import into the cell. And so if you have any sort of molecule like something that might inhibit autophagy, for example, like chloroquine, you're also going to impinge upon this process. So just an FYI, any kind of like baflomycin or something like that will also inhibit iron metabolism. I think that's something that's really overlooked when people do these sorts of experiments. Okay, so which downstream iron sulfur cluster containing protein is responsible for the effects on cell viability? So here's a list of known proteins that contain iron sulfur clusters. And you'll notice that a lot of these are involved in oxidative phosphorylation in the mitochondria, which is fairly interesting. But also you have a variety of proteins in the nucleus that include a lot of helicases that are involved in DNA repair, and then all three of the replicative polymerases. And so in our model cell line, we made a model in which we could express a doxycycline repressible cDNA and delete the endogenous copy of the gene to really ask what are the kinetics of loss of this protein in a cell line that are independent of RNAi. And so you can see that when we add doxycycline, we really don't need to add much to suppress gene expression, so it's not something that's going to be inhibiting the mitochondria. We get a fairly good suppression of expression of this protein over about a period of a week. And there were a lot of phenotypes that I'm going to describe, but one of the things that stood out the most was that nucleotide incorporation slowed and then eventually halted concomitant with loss of this protein. And the cells are rested in some weird sort of G2 kind of arrest. And we can blot other proteins that are indicative of what's going on here. So this protein, Pol delta, is a nuclear protein that contains an iron sulfur cluster, and this is also the lagging strand polymerase. And so you can see that the levels of this protein are known to be dependent on the presence of an iron sulfur cluster cofactor, and this protein slowly goes away right after you lose biosynthesis of the cofactor. You get an increase in this marker of replication fork stalling, check 1, and then afterwards you get an increase in this marker of double-stranded DNA breaks gamma H2AX. And so consistent with the stalling of replication that we see. One other piece of data that we collected around this time was that when we looked at this effect across different breast cancer cell lines, we saw that one subtype of breast cancer, this basal-like breast cancer subtype, was really strongly affected by inhibition of iron sulfur cluster synthesis, while other cell lines were affected to a lesser extent, suggesting that there might be some specific requirement for this biosynthetic pathway in this subtype of breast cancer. And basal-like breast cancer is something, or triple-negative breast cancer, that there's a lot of overlap between those two categories, have limited targeted therapies. And so finding novel targets for this type of cancer would be something of interest. And so we did a lot of work trying to understand which proteins that contain iron sulfur clusters might be responsible for this difference in sensitivity of luminal and basal-like breast cancer. And this is one piece of data that addresses that. So we looked at the DepMap data from the Broad Institute, and this is looking at genes that are essential in the same cell lines. So for example, genes that are cell lines that require ISCU for their viability, and ISCU is an iron sulfur cluster biosynthetic protein, also require ABCB7, and they also require NFS1 and frataxin, all members of the core iron sulfur cluster biosynthetic machinery. And so the same cell lines in the Broad data set, which is now, I think, several hundred cell lines, the same cell lines are hypersensitive to suppression of those genes. And that suggests that those cells are in the same biosynthetic pathway. And of course, we looked at proteins that contain iron sulfur clusters, and we noticed that DNA polymerase epsilon was had the highest level of correlation with all of these iron sulfur cluster biosynthetic genes, suggesting that this protein is an iron sulfur cluster containing protein that whose phenotype of suppression is underlying the iron sulfur cluster requirement in the cell. So what is DNA polymerase epsilon? I already mentioned that it's appreciated to be the leading strand replicative polymerase. I also introduced Pol Delta, which is the lagging strand replicative polymerase. There's also a primase, DNA polymerase alpha, that synthesizes off of the RNA primer to initiate replication. All three of these proteins contain iron sulfur clusters. And so we investigated each of them and suppressed them in our panel of cell lines. And we saw that indeed DNA polymerase epsilon suppression really had a dramatic effect on basal-like breast cancer cells and to a lesser extent on other cell lines. So all of these cell lines, I should say, require leading strand replicative polymerases to function. It's really just that at the level at which we can suppress it by RNAi, do we see a phenotype, a dichotomy between these basal-like breast cancer cell lines and these non-basal-like breast cancer cell lines. And this was an effect that we didn't see with the other replicative polymerases, so that this dichotomy didn't exist, suggesting that there's something special about leading strand replication. And not surprisingly, when we looked at a model of tumor growth in the flanks of immunocompromised mice in a DOX-inducible fashion, we see that blocking DNA polymerase epsilon blocks tumor growth. Perhaps not surprising given the in vitro data that I just showed you. And we looked across this panel of cell lines, not just at the level of DNA polymerase epsilon, which was suppressed pretty much equally well across the panel, but at two markers, one of fork stalling, this phosphorylation of CHEK1, and one of double-stranded DNA breaks, this phosphorylation of H2AX. And we saw that in all of the cell lines in which we saw a dramatic growth effect in all of these basal-like breast cancer cell lines, we got an increase in this fork stalling marker, and we got an increase in this marker of double-strand DNA breaks. We really saw only one outlier, this luminal breast cancer cell line, EFM19. And so this suggested that something about being a basal-like breast cancer cell line made you more susceptible to DNA damage and cell death downstream of blocking replication. And so what's going on there is a question that we wanted to ask. We also noted that this effect on suppressing leading strand replication was irreversible after about 2 days. So using this cell line in which we can suppress DNA polymerase epsilon, we would withdraw DOX after inhibiting replication. And really 2 days on DOX was sufficient to completely prevent the cells from recovering. And then we went on to ask which of these iron sulfur cluster cofactors that are present in DNA polymerase epsilon are required for its function. So DNA polymerase epsilon actually has two iron sulfur cluster cofactors, one in the polymerase domain and one at this C terminus next to this zinc finger site. And we made mutants of this DNA polymerase that lack each of these iron sulfur clusters. And the wild type enzyme was able to rescue the effect of suppressing the enzyme, not surprisingly, and as was the enzyme in which we had mutated the C-terminal iron sulfur cluster, but the enzyme in which we had mutated the N-terminal iron sulfur cluster binding site was unable to rescue, suggesting that this iron sulfur cluster was really key for the protein function. And I should mention again that the other two replicative polymerases, alpha and delta, have iron sulfur cluster cofactors. These cofactor binding sites are present at this C-terminal site next to a zinc finger, similar to epsilon, but they lack this N-terminal polymerase iron sulfur cluster binding site, which may explain why we see a difference in suppressing epsilon versus alpha and delta with respect to differential essentiality in basal versus luminal breast cancer. And so this is an interesting difference. Okay, so what happens, what other phenotypes do we see in these different cell lines when we suppress this biosynthetic pathway, or when we suppress DNA polymerase epsilon, because the phenotypes are overlapping? As I mentioned, for iron sulfur cluster synthesis, we see a loss of incorporation of nucleotides and arrest of cells in this weird G2M phase of the cell cycle. Interestingly, though, similar levels of suppression of the replicative polymerase reduce the level of nucleotide incorporation, But otherwise, the cell cycle is fairly normal, meaning that the percentages of cells in each of the phases is similar, and we don't see cells stalling with this weird intermediate level of DNA. We don't see a lot of cells in this sub G1 peak, suggesting that these other cells are able to sort of muddle along with low levels of replicative polymerases, and are able to sort of appropriately respond to having low levels of polyepsilon or of iron sulfur cluster synthesis. Another phenotype that we saw quite a bit in the basal-like breast cancer cells was these regions of single-stranded DNA. So sometimes when you inhibit replication, you get decoupling of the polymerase from the helicase. And if that happens, if the helicase gets ahead of the polymerase, you then have regions of single-stranded DNA. And so it seemed like this was happening to a large extent in these basal-like breast cancer cell lines, whereas the coupling was maintained potentially in these other cell lines that were not getting DNA damage when we inhibited leading strand replication. Something else that we found really interesting and that we're really curious to follow up about on is that polysuppression induced a senescence-like phenotype. And this is not something that we saw with inhibiting the other replicative polymerases. But this phenomenon of activation of these NF-kappa B targets and the appearance of these senescence-like markers suggested that inhibiting leading strand replication or iron sulfur cluster synthesis was really triggering a different phenotype than inhibiting lagging strand replication or the primase activity. And a lot of these NF-kappa B targets are interesting because this is induction of a cancer cell intrinsic secretion of cytokines and chemokines, which may be able to remodel the tumor microenvironment in an interesting way. And so a lot of current work is focused on trying to understand how inhibiting leading strand replication in basal-like breast cancer is activating NF-kappa B, and whether these cytokines or chemokines are having an effect on the tumor microenvironment, and then whether that effect is helpful or hurtful to downstream anti-cancer therapy. Okay, so here's the model of what I just described to you. So cells can have normal functional polyepsilon. We can induce a state where there's low functional DNA polymerase epsilon, either by directly inhibiting the protein or by inhibiting iron sulfur cluster synthesis in the context of high oxygen, so that we lose the cofactor, which is pretty much just as good as inhibiting the synthesis of the polymerase. This can lead to some kind of replication stress, and whether the cells are able to manage that replication stress seems to be dependent on whether they're in basal-like breast cancer or luminal breast cancer cells. Luminal breast cancer cells can maintain their viability, whereas basal breast cancer cells have these phenotypes, which I described. And so one really important question is, what's going on here? Like, why is there this difference? And so one key piece of data that we found casting about various hypotheses for why this was the case was that when we inhibited ATR or CHEK1, these two kinases that are responsible for sensing single-stranded DNA breaks or for sensing stalled forks, these could induce sensitivity to polysuppression in a cell line that normally is fairly insensitive. So this N is inhibiting NFS1 and this P is inhibiting DNA polymerase epsilon. And you can see that when we do this in MCF7 cells, we don't really get much of an effect, as I mentioned before. But when we layer on inhibition of these kinases that are responsible for sensing this type of DNA damage, we get a lot of DNA damage. And this isn't true for other kinases like ATM or DNA PK. We didn't see a similar effect. It was only ATR and its downstream effector CHEK1. And so ATR and CHEK1, as I mentioned, can sense DNA damage. CHEK1 has a variety of downstream effectors. Two very important ones are these CDC phosphatases that regulate cell cycle control by regulating inhibitory phosphorylation marks on CDK1 and CDK2. And these phosphorylation sites are the site that are phosphorylated by the Wee1 kinase. And so Wee1 inhibits CDK2 and CDC25A then relieves that inhibition and enables G1 to S in the case of CDK2 or the G2 to M transition in the case of CDK1. And so we wondered if this pathway was responsible for the phenotypes that we were seeing, knowing that inhibition of either ATR or CHEK1 would lead to activation of CDK1 and CDK2. Inhibition of ATR or CHEK1 would lead to activation of CDK1 and CDK2, whereas stimulation of ATR or CHEK1 would block cell cycle progression by inhibiting CDK1 or CDK2. We also noticed, interestingly enough, that in basal-like breast cancer, there were a lot of genomic alterations that would be expected to activate CDK2 either directly or downstream, whereas luminal breast cancer has genomic alterations that would act upstream on CDK4. And so we wondered whether or not this dichotomy might also be playing a role in the differences that we saw. Okay, so one of the first things that we did was that we looked at the effect of inhibiting or activating CDK2 or CDK1 using this Wee1 kinase inhibitor. And at the same time, layering on partial inhibition of CDK1 or CDK2. And so here, as was with the CHEK1 or ATR inhibitors, when we suppress DNA polymerase epsilon and also iron sulfur cluster synthesis, it will cooperate with this Wee1 inhibitor to cause DNA damage. But this can be completely rescued with a CDK2 inhibitor, but not so much with a CDK1 inhibitor. And we obtained similar data using ATR or CHEK1. And actually, that's what's shown here. So the CDK2 inhibitor can block this accumulation of DNA damage when we've downstream of ATR or CHEK1 inhibition, suggesting that it's really this CDK2 step that's responsible for what's going on here. Another piece of data that was fairly interesting was that we could look at phosphoproteomic data from human tumors. So these aren't cell lines, these are human tumors that are annotated as being basal-like HER2 luminal A or luminal B based on the PAM50 annotation. And we find that basal-like breast cancer cells actually indeed have hyperphosphorylation, a variety of CDK2 targets compared to these other cancer types, supporting the idea that there's dysregulation of this step in basal-like breast cancer. Okay, so to come back to what we think this difference is in basal versus luminal breast cancer cells, we think that when there's replication stress induced by iron sulfur cluster deficiency or low DNA polymerase epsilon, the cells respond to that based on whether they have normal or regulated CDK2 activity and can maintain viability, or based on whether they have high CDK2 activity, either because they have some genomic alteration that promotes that, or because we've pharmacologically intervened to induce a state where we have high or dysregulated CDK2 activity. And our working hypothesis is that some downstream effector of CDK2 is regulating the response to replication stress, and that when that's abrogated, cells will continue to replicate despite having replication stress, and that will lead to this, this DNA damage that we observe in basal-like breast cancer. And there's actually some precedent for that. There's a recent publication out showing that in a sort of an unperturbed cell cycle, CDK2 activity goes from being very low when cells are in G1 to being elevated when cells are in S phase to induce, again, the G1S transition, but also that during S phase, CDK2 activity can transiently be downregulated in response to replication stress or DNA damage that just sort of stochastically occurs when cells are proliferating. And so you get this state where CDK2 activity is elevated during S phase, and then it can encounter some kind of replicative stress. It will be downregulated transiently. And then when the DNA damage is repaired, then the CDK2 activity can normalize and the cell can continue replicating. And so we think that this ability to fine-tune CDK2 activity during S phase, also known as the intra-S checkpoint, is really important in understanding how cells will respond to a variety of things that include iron deprivation, inhibiting autophagy, which will affect iron metabolism in the cell, or directly inhibiting, in this case, DNA polymerase epsilon or leading strand replication. And as I just mentioned, there are a lot of ways in which that are relevant to cancer metabolism or things that a lot of people in the metabolism field care about that impinge upon these pathways. Anything that perturbs lysosomal pH will affect iron sulfur cluster metabolism, and anything that will, of course, inhibit nucleotide synthesis will cause replication stress. And so thinking about how iron metabolism and replication stress affect the cell, it's important to consider I think these effects on CDK2. And in particular, there's been some reports that treating cells with things like baflamycin really do inhibit cell proliferation because of iron, because if you can then rescue poisoning the lysosome by giving these sort of inhibitors of lysosomal pH, like baflamycin or chloroquine, by giving the cells extra iron, really suggesting that that's one of the key functions of the lysosome with respect to short-term viability of the cells, is providing iron for replication. Okay, so thank you for your attention. I'd like to thank the lab members who did this work, who are listed in bold here, as well as our collaborators at NYU and Rockefeller, as well as our funding. And I will be happy to take your questions. And I think I can hide this, maybe.
Great. Great. Thank you very much, Richard, for your excellent talk. From the guys from the audience, if you want to ask some questions, just put on the YouTube comments box, and we are going to address them to Richard. So Richard, it's very interesting your work, and one of the things that strikes me the most is it's because you have the modulation of the iron sulfur cluster only affects the polymerase epsilon, right? Mm-hmm. And the delta is not affected, and maybe it's due to that difference in their structure, right? So it means that the other sites, they are not very important for the polymerases.
So when okay, so the iron sulfur cluster site in epsilon in the polymerase domain, we think is important for the basal-like specific essentiality. The other sites are critical for the function of those enzymes, alpha and delta, in that if you delete them, the proteins become really unstable. And if you inhibit iron sulfur cluster synthesis, you also, again, lose the stability of those proteins. It's just that 95% or whatever inhibition of delta doesn't really give you much of a phenotype in cycling cells. And 95% inhibition of alpha gives you an effect that really can't be distinct. There's no distinction between basal and luminal breast cancer. So they're important for the function of the cells, but for some reason, it's the epsilon function that is something about inhibiting leading strand replication that is making the basal-like breast cancer cells more sensitive. We have new data that also goes to this point where if we completely delete delta, now we can really kill the cells. And the type of cell death is different than if we delete epsilon. So if we delete epsilon, the cells senesce, or they enter this senescence-like state where they just kind of like stop. If we delete delta, the cells explode. So there's something else interesting going on there, where if we really go to zero, the effect is different than if we, sort of inhibit 95%. Anyway, that's a lot of information to answer.
So it's very interesting. But if you inhibit the delta alone, not inhibiting the epsilon, right? You kill the cell. It's very interesting.
Yeah.
Because I was thinking about this difference between the lagging and the leading strands, right? And another question that, because you've seen this difference between the different types of breast cancer, breast tumors, right? The more aggressive and the less aggressive ones, and you have a different response between them, right? Do you think that it can be translated to other types of tumors?
Yeah, we've been thinking about other tumor types that have that are phenotypically similar to basal-like breast cancer. And the first thing that comes to mind is ovarian cancer. And we've seen some sensitivity to inhibiting epsilon in ovarian cancer, but I think it might be a bit more complex. And other than that, we haven't cast the net too wide, looking at other tumor types. We've been focused a lot on trying to understand what's going on downstream of CDK2, and then trying to understand this NF-kappa B signaling. That's most of where our focus is right now.
Nice, great. We have some questions here, and Gabriela is asking, excellent talk. Have IRP1 or 2 and then NFS1 expression levels been addressed in breast cancer patients?
Right. So most of the iron sulfur cluster biosynthetic machinery is upregulated in transformed cells. And this is fairly universal, presumably because you need additional iron sulfur clusters for things that are involved in proliferation, such as replication. And there are also some nucleotide biosynthetic proteins that contain iron sulfur clusters. And so the whole pathway is upregulated. It seems to be more upregulated when you're in a high oxygen environment, perhaps suggesting that there's some feedback control. With IRP1 and 2 levels, I don't think anyone has really looked at that, and I can't quite recall any data there. So I'm gonna have to think about that one.
There is the continuation here, and she keeps on saying, considering the association of response to ferroptosis with basal type breast cancer, I wonder whether there is any interplay between estrogen receptors and the ferroptotic molecular machinery.
That's an interesting question. There was a paper by Stu Schreiber suggesting that cells that have more of a mesenchymal character, which basal-like would be sort of adjacent to that, are more susceptible to ferroptosis. And the reason is because they have more polyunsaturated fatty acids in their lipid membranes. And that's really a driver of whether or not a particular cell type is going to be sensitive to ferroptosis, in addition to sort of this iron overload phenotype that we're inducing. And so if I were to look for that, which I really haven't, I would wonder whether or not estrogen receptor signaling was affecting the level of polyunsaturated fatty acids and lipid membranes. And there are some key genes that are involved in that process where you would have more or less polyunsaturated fatty acid content. But I'm not aware of those data offhand, but I think that would be an interesting place to look to whether or not estrogen receptor expression affected that.
Great. I have one last question. It's because since I work with brain tumors and we have this IDH mutation, which uses isocitrate, right? And isocitrate is a product from the aconitase, which is very important for the iron sulfur cluster metabolism. Have you ever seen some interplay between this IDH mutation and the iron sulfur cluster metabolism?
You know what? I haven't looked for that, and it's a really interesting question. So iron sulfur clusters are required for both the aconitase step, but then also for the alpha ketoglutarate dehydrogenase step. And so I think some of that is going to, so it kind of like bookends that little bit of the TCA cycle. And so it's quite possible that if you have a lot of glutamine anaplerosis into the TCA cycle, that you can then have some sort of effect on, say, alpha ketoglutarate levels or something like that, if you have some partial inhibition of iron sulfur cluster synthesis. But I know, it's always been something that I found really interesting is, if you're producing 2-HG, but through an IDH mutation, and you're sort of vacuuming out all of this alpha-ketoglutarate from the cell and sort of making it into some sort of terminal metabolite, how much of a stress does that really put on the cell? And you might imagine that in a cell that has an IDH mutation, it might be more dependent on iron sulfur cluster metabolism for that reason. I haven't thought about that. I guess, again, it would depend on how much glutamine anaplerosis you have. And so for maybe like a pancreas cancer cell line, which has a lot of glutamine anaplerosis, it might not affect it that much. But I'm not as familiar with how much glutamine anaplerosis there is in IDH mutant tumors. That might be something interesting to think about.
Great. Well, so I think we are going to end today, and I'm gonna ask for Alicia to come back. Hervé, he had to get out, but just to finish our first day, I have to thank you guys very much for your inputs here, and I think it was very important to be discussing here. Alicia, do you want to talk something about Richard speak or some question?
No, just a quick comment that IDH is a major source of NADPH, especially in the absence of NNT. So that might be something to look at also, as is glutamate dehydrogenase in mitochondria. It's
interesting because glutathione, which of course is going to be recycled from NADPH, is also important for iron sulfur cluster synthesis, and so there may be some interplay there that's interesting.
Yeah, okay. So guys, thank you very much. And I would like to remember you that tomorrow we will have our second day, right? So it will be at the same time at 9 a.m. from Brazil. And we are looking forward to seeing you, everybody here again tomorrow, okay? So thank you very much. Bye-bye.